What's going on, wonderful people? This is Medicosis Perfectionalis, where medicine makes perfect sense. In the previous two videos, we started talking about microbiology. Video number 1 was an introduction. Video number 2 was the gram stain technique and the difference between the gram-positive bacteria and the gram-negative bacteria. Today, let's dig deeper into the land of the gram-positive bacteria. Gram is a stain named after the doofus who discovered it. If a bacteria is gram-positive, it appears purple under the microscope when you use the gram stain. And then we have two types. You could be purple and spherical. We will call you a coccus. And the plural is cocci or cocci. Or you could look like a rod, and we will call you bacillus, and the plural is bacilli. One of those gram-positive cocci is Staph aureus, which can cause an abscess in your brain. With that said, now let's get started. Please watch these videos in order, and they are arranged in my microbiology playlist. Do you remember biology? Kingdom, phylum, class, order, family, genus, species. When I write Staph aureus like this, Staphylococcus is the genus. Aureus is the species. Be very careful. It has to be written in italics if you want it to be a bacteria or a fungus. So if I write something like this, Haemophilus influenzae, that's a bacteria because it's in italics. But if I write like this, no italics, this is referring to influenza virus. This is a bacteria, but that's the virus. Big difference. So I took a sample from the patient. The sample has bacteria, and then I put it under a microscope after staining my sample with gram stain. If it appears pink, it's gram negative. If you're positive, you could be cocci or rods. If you're negative, you could be cocci or rods. Okay, suppose that the bacteria is gram positive. It could be cocci or rods/bacilli. I am a coccus. Well, are you catalase positive or catalase negative? What does that mean? Do you possess this enzyme? Do you have it? Oh, I have this enzyme. Then you are catalase positive. No, I do not have this enzyme. Then you are catalase negative. Staphylococci are catalase positive, but Streptococci are catalase negative. What the flip is a catalase? A catalase ends in A, so it's an enzyme that catalyzes. Catalyzes what? The conversion of H2O2 into two things, H2O and O2. Of course, I will have to balance the equation, but you get the point. This is hydrogen peroxide, very toxic, but this is water and oxygen, not toxic. Okay, medicosis, I am gram-positive, I'm a coccus, I'm catalase positive, and then I can divide you into two groups. The ones that are coagulase positive and the ones that are coagulase negative. How do you divide streptococci? Based on whether they are alpha hemolytic, beta hemolytic, or gamma hemolytic. More on that later. How about the rods or the bacilli? Well, you ask yourself, are they spore forming or non-spore forming? Let's start with those who do not form spores. They could be aerobic, I need oxygen, or anaerobic, no air. I can live without oxygen, I don't need you. Moreover, the aerobic could be subdivided into motile and non-motile. Hey medicosis, I wanted to ask you a question. Is Staph aureus motile? No. We're done with these doofuses. Let's talk about the spore-forming gram-positive bacilli. Let's go. Again, aerobic or anaerobic. The aerobic, motile or non-motile. The anaerobic, motile or non-motile. Of course, all of these bacteria will be discussed in subsequent videos. But if you want quick review right now, go to my playlist titled Medicosis Picmonic, and you will find them in a very quick review. The detailed discussion will be found in this playlist called Microbiology. And now, on to today's topic, the gram-positive bacteria. I could be gram-positive cocci or gram-positive bacilli. Let's talk about the cocci. Based on whether you have catalase or not, I can divide you into catalase positive, staph, catalase negative, strept, and their friends. Every single stinking bacteria in this slide is, number 1, gram-positive. Oh, that makes sense, which means it appears purple when you stain it with gram. Next, they are spherical. They are cocci. Oh, that makes sense. And third, they lack endospores. What the flip is an endospore? We'll talk about that in today's video. Just hold your horses. Okay, medicosis, I am a cocci, I'm a gram-positive cocci, and I'm catalase positive. Then you gotta ask yourself, are you coagulase positive or coagulase negative? Oh, I'm coagulase positive. This is Staph aureus. I am coagulase negative. This is Staph that's not aureus. Example could be Staph epidermidis or Staph saprophyticus. And this distinction is based on what? Whether you are novobiocin sensitive or resistant. What is novobiocin? It's a medication. It's an antibiotic. Oh, it is supposed to kill the bacteria. Yes, if it kills the bacteria, it was epidermidis. If it did not kill the bacteria, i.e. the bacteria is resistant to this antibiotic, then we're talking about Staph saprophyticus. Hey, medicosis, why do we call them staph in the first place? Because they are clustered like grapes. Oh, this is what staph means? That's true. And what does aureus mean? It means golden, because their colonies appear yellow or orange. Why is that, medicosis? Because they have carotenoid pigments. This is why the carrot is yellow/orange. Okay, medicosis, forget those staph that are clustered like grapes. Tell me about the strep. Okay, they are like strips. What do you mean? They are arranged like this in cords or chains. Oh, I get it. All right, and then you put them on a dish and see their hemolytic activity. I could cause partial hemolysis to the agar or complete hemolysis or no hemolysis. Therefore, you are either alpha hemolytic, partial hemolysis, beta hemolytic, full or complete hemolysis, or gamma hemolytic, no hemolysis. If you're alpha hemolytic, it's one of two main possibilities. I'm not giving you every example. I'm giving you the most important and clinically relevant example. Okay, this distinction is based on what? Are you sensitive to uptoken or are you resistant to uptoken? Uptoken is another antibiotic like novobiocin. If it kills you, strep pneumo. If it cannot, strep viridans. This is the old name. Today they call them the viridans group Streptococci, or simply the viridans group of bacteria. So strep viridans is the old name. Next, I am strep, which means what? I'm gram-positive coccus arranged in chains, and I cause full hemolysis. Thank you. Based on your sensitivity, or lack of it, to bacitracin, we can divide you into group A, I am sensitive, or group B. Group B, kiss my butt, bacitracin. Where do you live, group B? I live near the buttocks or the pelvic area of a pregnant lady. And if the doctor is a doofus and did not catch me and treat me, I can ruin the baby's life because I can cause neonatal sepsis, neonatal meningitis, neonatal pneumonia. You better be careful of group B strep. For the next generation to be or not to be depends on whether the doctor is a buffoon or competent. Okay, medicosis, what if I am sensitive to bacitracin? This is group A strep. It's also known as group A beta hemolytic streptococci. What's the name of the species? Pyogenes. So Streptococcus pyogenes. Why do you call it pyo? Pus. Why gene? Because it causes genesis of pus. Oh, so it's gonna cause pus diseases? Yeah. On the skin? Yeah. Like sepsis, cellulitis, necrotizing fasciitis, impetigo, etc.? You bet. Or I could be gamma hemolytic, which means no hemolysis. Okay, now can you grow in salt solution? Yeah, I can grow there. Enterococcus. No, I cannot grow. The salt is so salty for me. It's gonna kill me. Strep bovis. Now, if you do not bring a blank piece of paper and draw everything here in detail in your own handwriting without looking, there is no hope for you. And do it twice. Again, what's common among every coccus in this freaking slide? Number 1, all of them are gram-positive. Number 2, all of them are spherical or cocci. Number 3, they cannot make endospores. Thank you. Quiz time. Can you differentiate between staph and strep without a microscope, without any stains, without cultures, and just by using one drop of a solution? Do you know the answer? Pause and try to think about it. Yeah, I can do that. I add a drop of any solution that has catalase in it. Good job. Catalase is going to do what? It's going to catalyze the following reaction. The conversion of hydrogen peroxide, the harmful, into water and oxygen, harmless. Oxygen can do what? Can make bubbles. Oh, so if I see bubbles, it was staph. No bubbles, it was strep. Why? Because staph is catalase positive. It is bubbly. But strep cannot do this. No bubbles. Easy peasy. Hey medicosis, is H2O2 good or bad? Well, there are no solutions in life. There are only trade-offs, as Dr. Thomas Sowell said. So, when it comes to the bacteria, H2O2 is good for you because it kills the ugly bacteria that's trying to kill you. Oh, I get it. So hydrogen peroxide is made by your body. From what? From oxygen. So you start with oxygen. Where do I get it? Like, remember your mitochondria? Yeah, you breathe oxygen in. Oh, okay, that's fine. And then you convert it into superoxide and then into hydrogen peroxide. All of these are called reactive oxygen species or free radicals, which is not a political movement. After hydrogen peroxide, you make hydroxyl free radicals, and then it depends. If you're talking about protecting your own body, just convert them back to water. But if you're talking about destroying the bacteria, then you can destroy it with H2O2, or you can make it worse for your enemy. Convert this hydrogen peroxide into HOCl, hypochlorite, which is basically bleach, and throw that bleach in the face of the bacteria and destroy it. But some bacteria are sneaky because some bacteria have what? Have catalase enzyme. Oh, they will take your poison, the harmful hydrogen peroxide, and convert it to something that's not gonna hurt the bacteria when you throw it in their faces. Oh, wow, that's the catalase. Yeah, it converted the harmful H2O2 into harmless water and oxygen and some bubbly action. This is the purpose of catalase. This is how your bacteria evade your defense mechanisms. Alright, medicosis, I remember you told me that all the bacteria that you enumerated in that stinking slide did not make endospores, right? Yeah. But can you tell me what are the endospores? Sure. Spore means what? A sphere like this. Oh, a shell. Alright. And where's the bacteria? Inside the shell. Endo. Oh, the bacteria has encircled itself in a bubble, in a strong shell to protect herself from unfavorable environmental circumstances, such as food scarcity. Notice I said food scarcity, not food shortage, because shortage is a price phenomenon. But that's a story in economics for another time. Back to microbiology. Can every bacteria make endospores? Shut up. Only some gram-positive bacteria make spores some of the time when the environment is unfavorable, when the conditions are dorky. But don't ever forget, gram-negative bacteria do not make spores. Never ever. And this is a famous question on your exam. However, gram-positive bacteria, some of them make spores, some of them do not make spores. Let's talk about the ones that make spores. Alright, I only make them when the environment is not good, when I have problems like food scarcity, when something is about to harm me, and when this bad thing happens, I will defend myself. I will convert myself from a vegetative state capable of dividing into a dormant state protected within a shell. And this shell is called the spore. Tell me about your shell. My shell is dehydrated. Okay. My shell is multi-shelled. It's not one layer. It's gazillion layers. My shell is protective. And this lovely spore is made of what? Calcium bound to dipiclonic acid. Hey medicosis, what's the difference between me boiling my soup at 100 degrees Celsius and the microbiology lab boiling at a higher degree known as the autoclave? Is this better? Sure, because your boiling is awesome, but it cannot kill everything. It cannot kill some spores. However, the autoclave can kill more spores than you. Oh, I get it. So the spore is really protective. Even after boiling your food. Oh, I'm cooking it. I'm destroying all the bacteria. Shut up. You're destroying many bacteria, but not all of them. Be very careful. Here is a very important concept. Remember that staph is coagulase positive, but strep, especially strep pyogenes, is coagulase negative. Why is this important? Coagulase, what's that? It's an enzyme that coagulates. Oh, it makes fibrin fibers? That's true. So it converts fibrinogen, which is not active. It's a zymogen. It causes genesis of fibrin. Oh, but when it's fibrinogen, it's not active yet. Once you convert it to fibrin, this is the active form of the protein. And what's fibrin? Some fibrous fibers. And what do? They protect the Staph aureus. Oh, it's like a small shell. Yeah, but this shell did not belong to the bacteria. This was your fibrin. This was your fibrinogen. The bacteria took your fibrinogen, converted to fibrin, and now the bacteria is hiding from your immune system. And that's why the infections caused by staph are usually localized. Oh, because they are hiding inside the fibrin shell. So we're talking folliculitis, teeny tiny and well localized. Abscess, slightly bigger but still well localized. Make it a little bigger, furuncle. Make it a little bigger on the back of my neck or on my back, carbuncle. But look at Streptococcus. It does not have a coagulase. It cannot hide within some lovely fibrin focus. That's why skin infections caused by strep are more widespread, more diffuse. We're talking sepsis, cellulitis, necrotizing fasciitis. Is this localized? No. Look at the abscess. Oh, that's a small thing. But look at freaking cellulitis. My goodness gracious. Necrotizing fasciitis is even worse. If this escapes and goes to the blood, sepsis or septic shock. Streptococcus can also cause erysipelas, which can confuse you with cellulitis. But notice there is a big difference between erysipelas and cellulitis. Erysipelas has look at this, look at the word, clear demarcations. You see this? Clear demarcations, and the lesion is elevated off the surface of the skin. Cellulitis does not have clear demarcations, and it's not as elevated as erysipelas. Cellulitis is more flat, erysipelas is more elevated and well demarcated. So if Staph aureus caused an abscess, can the surgeon remove it via incision and drainage? Yeah, just remove this teeny tiny spot. Okay, but can I remove a teeny tiny spot if you have cellulitis? Oh, heck no, because cellulitis is not a teeny tiny spot, it's a very wide area. And if this is so bad, you will not need incision and drainage. You will need skin grafting. Woo, this is serious. How about impetigo medicosis? Impetigo is in the middle because it can be caused by staph or by strep. And why did you draw it in this orangey brownish color? Because the description of impetigo is brown, crusty lesions on the skin. See, medicine makes so much sense once you understand what the flip you're talking about. Try to make it clinically relevant instead of just memorizing your textbook like a freaking donkey. But I don't blame you. It's your professor's fault. Pause and review and draw from scratch. Practice makes perfect. Repetition is the mother of pedagogy.
Medicosis, please help me. I have a collection of pus known as abscess.
Oh, I have good news of bad news.
What's the good news?
The abscess is confined to one area thanks to the coagulase of staph. Alright, therefore we can remove it easily. This is called incision and drainage. Alright,
what's the bad news then?
If this abscess is in your brain, it's also confined to a singular area. It can press on one structure, but we're talking about the brain here. If this presses on an area in your brainstem, my goodness gracious. As Dr. Thomas Sowell said, there are no solutions in life, only trade-offs. Let me tell you about exerting pressure in your brain. Do you remember physics?
Yeah.
Pressure equaled force over area.
Oh, this feels like eternity.
Now, do you have a car? No, medicos, I do not. All right, go to your uncle who has a car. Okay, and then you'll do two things. Metaphorically, do not try this in real life. In the first scenario, you will try to just clean the window. Just clean the windshield and wipe it clean. All right. In the second, do not do this. Punch the windshield in the face. Oh, what's going to happen? It's going to break. Why didn't it break here, but it broke here? Even though you were applying pressure, you were applying force in the first scenario and in the second scenario. What's the difference? Two differences. Number one, when you punch it, only a couple of knuckles hit the glass. Therefore, the surface area is less, which means the pressure is higher. the pressure is confined onto a smaller surface area, giving you more destruction. But there is another reason, and this is the angle. When you were wiping the windshield, you are massaging it. What's the angle between your hand and the horizontal axis?
No angle, it's 0.
Yes. And what is sine 0? 0. When you multiply 0 by something, what do you get? 0. Therefore, you will not break the glass while wiping it. Don't worry. But if you punch the windshield head on, what's the angle now between your hand force and the horizontal axis?
It's 90-degree medicosis.
What is sine 90, please?
1.
Oh! You will have the entire weight of your force on that spot of the glass, and you can break it this way.
But hey, medicosis, the difference between 0 and 1 is not that huge.
Oh, shut up. Just go talk to any software engineer. #learn to code. Now we will replace your fist, which was confined on a smaller area, with an abscess. An abscess is also confined to one specific area in the brain. When the surface area is low, the pressure is high. I can press on a structure in your brain, and this is called the mass effect. Two things will happen. Remember that your skull is like a box that is 100% packed and stacked and full. There is not an empty inch in your skull. Everything is packed to the fullest. Therefore, any abscess that's going to emerge that was not there before is going to do what? Is going to increase the pressure in your brain, and this is called increased intracranial pressure, aka intracranial hypertension. Can I get vomiting? Yeah, it's going to be projectile vomiting, too much pressure. Can I get headache? Yeah, usually frontal headache. That's worse in the morning. It gets worse when you lean forwards. Don't forget that your eye is part of your brain, and your cranial nerve is not a peripheral nerve. It's actually part of the brain. That's why multiple sclerosis is demyelination of the central nervous system. Also, it affects the optic nerve because the optic nerve is part of the brain. The eye is part of the brain because during embryology, literally your eye bulged forwards out of the brain tissue. Your eye belongs to the brain. And therefore, if you have intracranial hypertension, you know what else is going to happen? The pressure in your eye will increase. We call this papilledema. See how this mass effect is destructive? And it can cause focal neurological symptoms. You could have some motor deficits such as paresis, which is weakness, or paralysis, which is just you cannot move. Not weak, I just can't move. Sensory, it could be paresthesia, pins and needles, tingling, numbness, etc. Or it could be no sensation, anesthesia. Speech difficulty, slurred speech. Vestibular problems, I am dizzy, I have vertigo. Visual problems, I see double. See what happens when you integrate knowledge, unlike your woke professor with his PowerPoint? Because medicine makes so much sense once you understand what the flip you're talking about. In the next video, we'll talk about Staph aureus and the story of the chef who puts his fingers in his nose. Staph aureus is gram-positive, catalase positive, coagulase positive. Since it is staph, it is clustered like grapes. Since it's gram-positive, it appears purple. It can cause all kinds of infections. But take streptococci, like strep pyogenes, for example. Gram-positive, true. Cocci, true. Catalase, negative, no bubbles. Coagulase, negative, I cannot confine myself in an abscess, therefore I will spread. Sepsis, cellulitis, necrotizing fasciitis, and don't forget erysipelas. Let me prepare you for our discussion on Staph aureus by looking at this lovely picmonic. You can go to picmonic.com/VIP hookup/medicosis to see this in an animated video. Alright, Staph aureus, here is a staph of Oreos. Is a gram-positive, here is an angel. Coccus, spherical. Catalase positive, here is the positive cat. Catalase converts H2O2 into H2O and oxygen. Oxygen is bubbly. Coagulase positive. Staph aureus is beta hemolytic. Staph aureus secretes something called protein A. So remember staph, protein A. It's very virulent.
Why medicosis?
It inhibits your immune system. It inhibits your phagocytes. What you have seen is just one slide from picmonic, Staph aureus, but if you go right now, they have all kinds of bacteria. Picmonic is the best thing that happened to microbiology since Louis Pasteur. Go to picmonic.com/VIP hookup/medicosis and they will hook you up. Please pause and review and draw from scratch. You're watching my microbiology playlist. If you want an antibiotic series of lectures, I have 40 videos on my website, medicosisperfectionalis.com. This course comes with 40 videos, the notes in PDFs, 70 questions with answers, of course, 35 cases, and my Perfectionalis Ultimate Notebook, plus a mind map to help you memorize. Please print my pun and use it while watching the videos. I will see you in the next video when we talk about staph. In the meantime, please subscribe, hit the bell, and click on the join button. You can support me here or here. Go to Picmonic for animated medical mnemonics. Go to my website to download my premium courses like my antibiotics course. Be safe, stay happy, study hard. This is Medicosis Perfectionalis, where medicine makes perfect sense.